Hello, I'm Songyun Li, the postdoc of Lingang Laboratory in Shanghai. I'm glad to share our paper with you. The upcoming animation will present its key content. How CRH neuron in the lateral hypothalamic area regulate appetite in response to the food cues.
In our daily lives, we all understand that hunger can make even ordinary food more tempting. After we're full, though, these same foods lose their appeal except for particularly tasty ones. This is why we often see our colleagues snacking on various treats and fruits after lunch at the office. It highlights a fundamental fact. Our response to food cues is influenced by our physiological state and the tastiness of the food, leading to varying levels of appetite. So, which brain regions and neurons regulate appetite in response to food cues? Many brain regions have been identified to respond to food cues. For instance, in the hypothalamus, neurons expressing pro-opiomelanocortin in the ARC, inhibitory neurons in the DMAGE, excitatory neurons in the LHA, neurons expressing tachykinin in the PSTN, neurons expressing glucagon like peptide 1 receptor in the PVN can be activated by food cues. However, artificially activating these neurons either inhibits feeding or has no effect. In contrast, neurons expressing agouti-related protein, which promote feeding, show reduced activity when animals perceive food cues. Thus, scientists have long been uncertain about which group of neurons, under physiological conditions, promotes feeding behavior induced by food cues. In 1976, researchers discovered that the lateral hypothalamic area, known for its crucial role in feeding behavior, is activated by food stimuli in monkeys. We've identified that neurons expressing corticotropin-releasing hormone, CRH, in the lateral hypothalamic area are likely involved in this process. Unlike CRH neurons in the PVN, these are inhibitory neurons in the LHA. When 24-hour fasted mice eat, CRH neurons become notably active and their activity decreases when eating stops. Additionally, if 24-hour fasted mice eat continuously for 2 hours, the activation of these neurons when feeding gradually decreases. Moreover, when fasted mice are sniffing food, CRH neurons show a heightened response compared to well-fed mice. This means the same food stimuli elicit different feeding responses in CRH neuron reactions in various physiological states. When satiated mice are presented with delicious chocolate, whether they eat it or simply smell it, the activity of CRH neurons significantly increases. Simultaneously, if well-fed mice are injected with appetite-inducing ghrelin, they not only consume regular food but also regain responsiveness in CRH neurons that were previously unresponsive when they were exposed to regular food. When inaccessible food is presented, CRH neurons exhibit a significant response when fasted mice either remain near the food or attempt to access it. This suggests that the heightened activity of CRH neurons is not dependent on the occurrence of feeding behavior but is likely induced by sensory food cues. Furthermore, when fasted mice can only smell an empty container, the scent of food, or the scent of chocolate, CRH neurons show a notably higher level of activation in response to more appealing food odors. This indicates that the olfactory stimulus of food alone is sufficient to activate CRH neurons. To pinpoint the stage of CRH neurons activation accurately, we train mice to press a lever to obtain food, dividing the process into 4 stages: lever press, moving, approaching, and consumption. During the approaching stage, where mice approach the food, CRH neurons exhibit higher activity, compared to the lever press stage, representing food seeking. In other words, CRH neurons are most active when mice are approaching food.
While this group of CRH neurons exhibit similar responses to food as the neuronal clusters mentioned earlier, our subsequent findings indicate that there are notable differences in their impact on feeding when the neurons are stimulated.
Both optogenetic and chemogenetic activation of these neurons quickly induce selective feeding behavior in satiated mice, distinct from the non-selective feeding induced by VGAT-positive neurons in this region. Consistently, optogenetic inhibition of CRH neurons reduces feeding behavior in food-restricted mice, and this inhibition leads to a rapid cessation of feeding behavior when food-restricted mice begin eating. Furthermore, inhibiting CRH neurons when approaching food reduces the feeding frequency in food-restricted mice and decreases the preference of fasted mice for the scent of food compared to empty dishes. This suggests that inhibiting CRH neurons in a hungry state likely diminishes the attractiveness induced by food stimuli, leading to reduced feeding behavior. For long-term inhibition, mice expressing KIR 2.1 and CRH neurons, which can reduce neuronal membrane potential, experience slower weight gain. Clearly, CRH neurons can synthesize and release the specific neuropeptide corticotropin-releasing hormone, CRH. Therefore, we aim to investigate whether CRH peptide is involved in feeding behavior. After expressing CHR2 and CRH neurons, CRH shRNA was used to knock down the expression of the gene. We observed a significant reduction in feeding time when optogenetical activating CRH neurons, with the onset of feeding behavior also significantly delayed. Regarding neural circuits, we found that only optogenetic activation of the terminals of CRH neurons in the VTA, ventral tegmental area, and LC, locus coeruleus, but not LS, lateral septum, or PVT paraventricular thalamus, can increase feeding time and reduce the latency to initiate feeding in mice. Conversely, only inhibiting the terminals in the VTA can reduce feeding behavior in food-restricted mice and extend the time to initiate feeding.
Though our research has shed light on the aforementioned aspect and advanced our understanding how food cues influence animals' appetite through a modulation of CRH neuron activity, there is still a substantial journey ahead to gain a comprehensive understanding of the mechanism underlying brain's feeding-related systems.